Welcome students. Today I'll be discussing about nucleic acid, which is part of the chapter molecular basis of inheritance. So we know that nucleic acid was first isolated by Friedrich Mischer in 1869. The name nucleic acid was given by Altman in 1889 because it is found in nucleus and it is acidic in nature. Nucleic acid are of two types, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Now, the basic unit of nucleic acid is nucleotide. So the basic unit of nucleic acid is nucleotide. So now we will go through what is nucleotide made up of. Nucleotide is made up of three molecules. Number one, pentose sugar, which is 5 carbon compound, and it is also of two types, ribose sugar and deoxyribose sugar. Ribose sugar is found in RNA and deoxyribose sugar is found in DNA. In ribose sugar, the carbon 2 position of ribose sugar consists of hydroxyl group, whereas in deoxyribose sugar, the carbon 2 position does not consist of the hydroxyl group but the hydrogen. The next molecule is nitrogenous bases, which are of two types, purine, which is double ring structure, and pyrimidine, which is single ring structure. Purine are of two types, adenine, written as A, guanine, written as G. Pyrimidine, it is of three types, cytosine, thymine, written as T, uracil, written as U. That means all total, if when we say of nitrogenous bases, there are 5 nitrogenous bases, adenine, guanine, cytosine, thymine, uracil, and they are made in two groups. Those which have double ring is grouped under purine and single ring are grouped under pyrimidine. In DNA, the nitrogenous bases present are adenine, guanine, cytosine and thymine, and in RNA, the nitrogenous bases present are adenine, guanine, cytosine and uracil. Again one thing, when the polymer, that is the nucleic acid, is formed, so sometimes the nitrogenous base, they form the bond between them. Now always the nitrogenous base which forms the bond is always the purine bond, always forms the bond with pyrimidine. That is, always the adenine will bind with thymine with 2 hydrogen bond, or adenine with uracil with 2 hydrogen bond, guanine with cytosine with 3 hydrogen bond. Here, thymine may bind with adenine, uracil with adenine, cytosine with guanine, and vice versa. Now, the third molecule present in nucleotide is phosphoric acid, which supplies phosphate group for the formation of polynucleotide chain. That means the two nucleotides, they are joined together by this phosphate group. Now, how the polynucleotides are formed, we'll just check it out once. The pentose sugar, it joins with nitrogenous base to form nucleoside, and this nucleoside, when it gets added with the phosphate group, it forms nucleotide, and in this way the polynucleotide chain will be forming nucleic acid. Once how the polynucleotide chain is formed, once I will show it. This is the pentose sugar, 5 carbon pentose sugar. Always in carbon 1 position, the nitrogenous base, they bind with the pentose sugar at carbon 1 position. So I'll take it here, let it be adenine, and this is the nucleoside. When this nucleoside get added with a phosphate group in the carbon 5 position, it becomes nucleotide. So again, one nucleotide, I'll just show it. For example, guanine. So this is one nucleotide, this one nucleotide. Now I want to join the two nucleotides, and in this way the polynucleotide chain will be formed. So the two nucleotides, they join at this position. The carbon 3 position of one nucleotide and with the carbon 5 position of other nucleotide, the phosphate which will form the two ester bond. This is one ester bond and this is one ester bond, and they will join the two nucleotide. This bond is called phosphodiester bond or bridge, and the bond which forms between the pentose sugar and the nitrogenous base is called the glycosidic bond, and in this way the chain of nucleotide is formed, and later on they form the polymer nucleic acid. So this is all about nucleic acid, and in the next lecture I will discuss about the structure of DNA. Thank you.